Four major macromolecules in our body are carbohydrates, lipids, proteins, and nucleic acids. In this video, we will discuss proteins. Proteins are polymers that are made up of small repeating subunits called monomers. In this case, the monomers are called amino acids, which are joined together by a covalent bond called peptide bond. Now let's look at the structure of an amino acid. We have a central carbon atom that is attached to a carboxylic acid group at one end and an amino group at the other end. That is where it gets its name amino acid. In addition to that, the central carbon is also attached to a hydrogen atom, while the fourth group varies in different amino acid. If the fourth group is also a hydrogen atom, then the amino acid we have is called glycine. If the fourth group is an amino group or CH3, then the amino acid is called alanine. And if the fourth group is like this, then the amino acid we have is called valine. And if we add another CH2 group to valine, then it will be a different amino acid called leucine. Our body has 20 different amino acids that join together in different combinations and lengths to form thousands of different proteins in our body. For example, this is a very small protein made up of only 6 amino acids: glycine, alanine, valine, proline, leucine, and isoleucine. If only one of these amino acids is replaced with another one, then the structure and function of this protein will change. And if more amino acids are replaced, then it will become a totally different protein. Now let's see how these amino acids combine together to form long chains. When two amino acids come close, the carboxylic acid group of one amino acid reacts with the amino group of the adjacent amino acid. A water molecule is released, and the carbon and nitrogen of the adjacent amino acids are joined together through a covalent bond. This bond is called a peptide linkage or a peptide bond. You can see that we still have a free carboxylic acid group at one end and an amino group at the other end, which means that more amino acids can be added on either side to further elongate the chain. Now, all the proteins in our body are not present in the form of straight chains. In fact, there are four different levels of organization of proteins. They are primary structure, secondary structure, tertiary structure, and quaternary structure. First of all, let's discuss primary structure. It simply deals with the sequence of amino acids in a protein chain. For example, this very small protein is made up of amino acids like glycine, alanine, proline, and so on. This sequence of amino acids is stored in our DNA, which we will discuss later. If a protein is made up of 2 to 50 amino acids, it's also known as a peptide chain. And if it is made up of more than 50 amino acids, then it's called a polypeptide. The sequence of amino acid in a protein chain is extremely important. For example, hemoglobin is a protein present in our blood that binds to oxygen and carries it to different parts of our body. It is made up of four polypeptide chains, 2 alpha and 2 beta. Each alpha chains contain 141 amino acids arranged in a specific order, and each beta chain is composed of 146 amino acids also arranged in a very specific order. So in total, the hemoglobin molecule is made up of 574 amino acids arranged in 4 chains in a very specific order. The seventh amino acid in each beta chain is glutamic acid. It has a structure like this. In some cases, this glutamic acid is replaced by a different amino acid called valine. The normal shape of our red blood cells is biconcave like this. But due to this change in only one of the 574 amino acids, the shape of the cell becomes sickle-shaped like this, due to which the hemoglobin fails to carry sufficient oxygen to different parts of the body, which drastically reduces the life expectancy of people with this condition called sickle cell anemia. It also causes other symptoms like shortness of breath, dizziness, abdominal pain, and many more. Then we have secondary structure. The proteins are not always present in the form of straight chains of amino acids. In fact, they form different shapes like alpha helix and beta pleated sheets. We know that proteins are made up of amino acids that have an amino group and a carboxyl group that are joined together to form peptide bonds. Sometimes the polypeptide chains are oriented in a way that amino group of one amino acid is aligned with the carboxyl group of another amino acid, which is 3 to 4 units apart. These two groups are joined together through hydrogen bonding. These hydrogen bonds keep the shape of the polypeptide chain in the form of a helix. Each turn of the helix contains 3 to 4 amino acids. Sometimes these weak bonds are formed between the partial positive nitrogen of the amino group and the partial negative oxygen of the carbonyl group. These types of bonds give the shape of pleated sheets to the polypeptide chains. Then we have the tertiary structure. The overall folded three-dimensional structure of a polypeptide chain is known as tertiary structure. Sometimes it may also involve alpha helix and beta pleated sheets. In the secondary structure, the hydrogen bonds are formed between the amino group and the carbonyl group of the polypeptide backbone. But in the tertiary structure, the R groups or the side chains of the amino acids are also involved. For example, there is an amino acid called valine with a structure like this, and another amino acid called isoleucine with a structure like this. The side chains on both of these amino acids are non-polar or hydrophobic in nature. Such amino acids with non-polar side chains tend to stay together in a peptide chain, forming hydrophobic interactions. Then there are some amino acids like asparagine and glutamine whose side chains are polar but uncharged. These types of amino acids tend to stay together in a polypeptide chain by hydrogen bonding. Cysteine is a unique amino acid with a sulfhydryl or SH group in its side chain. When two cysteine amino acids come close in a polypeptide chain, the two SH groups react together in the presence of oxygen to give a water molecule. The two sulfur atoms are then joined together by a covalent bond forming a disulfide bridge. All these type of interactions maintain the 3D structure of a polypeptide chain in a functional protein molecule. Finally, we have the quaternary structure. Sometimes two or more polypeptide chains come together and form aggregations to form a single protein. For example, insulin is a protein-based hormone. It is made up of two polypeptide chains, one of which has 21 amino acids while the other has 30 amino acids. Both these chains stay together due to formation of disulfide bridges between cysteine amino acids within the same as well as different chains. When I was a student myself, I used to get this one question wrong every single time. The question is, hemoglobin has a dash structure. I used to get confused because hemoglobin is always discussed on the primary structure to emphasize the importance of sequence of the amino acids in a polypeptide chain. But the fact that hemoglobin is made up of four polypeptide chains means that it has a quaternary structure. Another recurring question is, dash amino acid forms disulfide bridges. The answer is cysteine. On the basis of structure and function, proteins are divided into two main categories, fibrous and globular. Fibrous proteins are thread-like in appearance. At most, they have secondary structure of organization, meaning that they sometimes contain alpha helices and beta pleated sheets. Fibrous proteins are insoluble in water and they mostly have a structural role in our bodies. For example, our hair and nails are made up of fibrous proteins. And another important example is silk, which we obtain from silkworms. Silk is also a fibrous protein. Globular proteins are spherical or globe-like in shape. These proteins have tertiary or quaternary structure. Globular proteins are soluble in water and they mostly have functional roles in our body. The most important example of globular proteins are enzymes, which carry out all the metabolic activities in our body. Another important thing to note is that excluding water and fats, our bodies are almost entirely made up of proteins. For example, our hair, nail, and skin is made up of a protein called keratin, which is a fibrous protein. Our bones are made up of another protein called collagen. Our muscles are made up of proteins called actin and myosin. And basically, everything that we can see in our body are muscles. And when we eat meat like chicken, beef, goat, etc., we say that we are eating a protein rich diet. That's because we are eating their muscles, which are made up of actin and myosin. Our organs like heart, lungs, and stomach are also muscles, which are made up of actin and myosin. The defense proteins called antibodies, which fight with the foreign invaders in our body, are also protein in nature. Then we have hormones, which carry out different physiological functions in our body. Some hormones are made up of lipids or steroids, while others are made up of proteins. The most common example of protein-based hormone is insulin, which regulates the level of sugar in our bodies. So in short, everything in our body is made up of proteins, which in turn are made up of specific sequence of amino acids. The sequence of amino acid is stored in DNA. That's why we say that DNA is the blueprint of our body or life in general.